Okay, so today we'll conclude this three-piece series of lectures on quantum biology. We'll try to bridge the scales. I talked a lot about physics, a little bit about quantum chemistry, and some problems linking between fields. Let's go into the biological systems and try to understand how we can connect different, not only scales, but also physical theories or formalism that are applicable to these different scales. That's one of the problems with biological systems, because they, the details matter, but also the overall structures matter. So you have to understand both the small and the large. And here you have a kind of overview of the scales, dimensions which matter from atomic, where we clearly know quantum physics is the appropriate tool to understand atomic and molecular structures, to molecules, small molecules, tens of atoms. Quantum chemistry has been clearly demonstrated to help us understand those. Then we go into biomolecules such as proteins and enzymes, and they have thousands of atoms as parts of their structure. A typical protein will have a few hundred amino acids. Each amino acid has something like 15 atoms or so. And that will lead to thousands or sometimes tens of thousands of atoms comprising a biomolecule. And these biomolecules also form larger structures such as filaments or complexes, actin filaments, microtubules. And each of these structures may contain thousands of individual proteins, for example. So you'll be talking about hundreds of thousands of atoms, up to millions of atoms comprising one integrated system. So that poses a big challenge how to describe it. And so we are entering into this gray zone, which we call mesoscale, where probably quantum effects will describe only a minority of behavior or some special degrees of freedom. The question is which ones. And I'll show you one example today where we can understand a little bit about that. And then when you go to a larger scale, such as single cell, that of course contains the membrane, all the organelles and the nucleus, everything, DNA, everything in it, then we are in a quandary in a way, because it's a small system thermodynamically. It's out of equilibrium because it's constantly consuming energy, as we discussed briefly last time. And below, a bit beyond that, we go into the organismic scale, where we talk about 10 to the 20th atoms or more. And typical descriptions will be at the microscopic level, thermodynamics, cell mechanics. So classical physics adapted to biological systems, describing them reasonably well, with some difficulties sometimes, but most of the time not. And that is not to say that these quantum effects do not matter at this scale. As you recall from the very beginning in physics, there was kind of a similar situation, that a lot of microscopic systems were understood very well, but with some specific features, like in blackbody radiation or specific heat of solids, there were wrinkles that couldn't be ironed out with classical thinking. So I'm not saying that because we just understand most of it at the level of an organism, such as a human being or a fly, it means that there's nothing to be learned by understanding it, looking for quantum effects. Okay, now, in addition to the size scale, there's also a very important energy scale. So in physics and quantum chemistry, we like to talk about the energies for each of the bonds that are made. So biological systems are made by forming bonds and putting together atoms into molecules, molecules into biomolecules, biomolecules into complexes, complexes into cells, etc., as you saw on the previous slide. So physicists like to understand things using the concept of energy or force, or torque sometimes. Chemists, on the other hand, or in biochemists in particular, tend to understand things better using kinetic coefficients. So what are the strength of interactions in terms of the rates of reactions. So how many reactions per second. But these two are related, so strong bonds translate into fast reactions, weak bonds into slow reactions. So that's the translation here. But from my point of view, I just want to look at these numbers. A lot of numbers here. I'll just point out to two, which are of great importance to understand biology from this perspective. The first one is, well, you have this kind of decreasing scale of energy values. So the covalent bonds are the strongest, about 90 kilocalories per mole. Remember that these bonds, of course, the strength of the bond depends on the distance between the interacting molecules. So this is at 1.5 angstroms. Ionic bonds about 60, disulfide 40, salt bridges 4 to 7 kilocalories per mole. By the way, that's also a biochemical energy unit, which probably, and also in cell biology you're familiar with. We'll switch to physical in a second. But the weakest of these bonds is hydrogen bond and van der Waals bond. So they are at about, so hydrogen bonds, the weakest are about 0.5 kilocalories per mole, typically probably in the mid-range 5 or 6 kilocalories per mole, and van der Waals 1 to 4. Why is this important? So these are bonds which biomolecules tend to favor because are weak enough to be broken if needed, but strong enough to confer some kind of stability against what? Well, against thermal fluctuations, because as we mentioned a few times before, thermal fluctuations, that is due to the fact that biological systems prefer to live in warm places, so room temperature to physiological temperature, by physical standards, very warm. That carries a lot of fluctuation dynamics. So collisions between gas molecules or water molecules occurring all the time and bumping into biomolecules. You don't want these accidental collisions to break up the structure, right? So you can compare it on the energy scale to something called kT, the unit of thermal energy. This is per degree of freedom. So anything that has the ability to move or rotate has that much energy for free due to the surroundings constantly bombarding it and giving it that kick. So that's 0.6 kilocalories per mole. As you can compare it, this will be on par with the weakest of the hydrogen bonds. So you can sometimes occasionally break a hydrogen bond by thermal fluctuations, but most of the hydrogen bonds will be at 1 or higher kilocalories per mole, which will assure that even the weakest of the bonds stay in place in the face of constant thermal fluctuations. As you know, if you heat up the biological system such as DNA or protein, or a protein up to 40 degrees Celsius or higher, this will cause denaturing. Denaturing means unraveling of the structure, and that's why this is due to thermal energy causing the breakage of these bonds. So we are just at the sort of fine boundary between detrimental effects and useful. We need thermal energy for enzymatic catalysis, some important reactions to be able to proceed, but we don't want too much of the thermal energy to destroy the structure in place. And as you know, high fever at this level could be fatal because of the damage to the DNA and the protein structure at these temperatures of 40 degrees Celsius or higher. Okay, so keep in mind kT. So anything which has an energy below kT can be dismissed as noise. In other words, useless. It will not make any change, any effect. Anything that is above kT will be difficult to destroy spontaneously or be disintegrated spontaneously. And the second most important number to remember, so this is one, try to remember, if you can, it's not a lot to remember, 0.6 kilocalories per mole. But anyway, the second number that is important in this connection is something which we call the quantum of the biological energy quantum. And that's the link to this, the topic of these lectures, quantum biology. There is such a thing as quantum of energy in biology. All biological systems produce and consume energy, consume nutrient, glucose, fructose, etc., proteins, and convert it into the energy of electrostatic energy of the phosphate bonds in the ATP and GTP molecules. And that quantum of energy, it ranges, depends on the conditions, the concentrations, etc., whether it's GTP. GTP has a bit less energy, maybe 10 times than ATP, but nonetheless, that's the range from 3 kilocalories per mole to 60 kilocalories per mole is the range of the quanta of energy that biology uses. It's a universal quantum. There's really almost no exception to this. And I will not talk about exceptions. There are some. You can create energy by separating charges, such as across the membrane or other surfaces. And, but anyway, why is this important? As you can clearly see, it's safely at least 5 times greater than kT. So that is kind of a sacred, protected amount of energy that can be recycled, delivered. And what is it used for? Well, GTP, for example, is necessary for the formation of microtubules. Each dimer of tubulin, which makes up microtubules, requires 1 GTP molecule to be so-called assembly competent. In other words, it cannot bind to a microtubule unless it has that quantum of energy. It's an interesting non-equilibrium process. You can call it energy pumping, just like laser. You have to pump that molecule with energy, and when you do that, then it can be carried to its destination and form a filament. When it, on the other hand, disassembles from the filament, is removed from it, it loses that energy. That energy is dissipated into the environment and converted into heat. ATP is the unit of another universal unit of biochemical, biological energy, and that unit, by the way, is necessary for any sort of directional movement, any work. You know, physics work is force times distance, right? Force times distance equals work. That means if you want to move any object against a force, let's say friction or viscosity or gravity, any force opposing it, you need to apply a counterforce, and the amount of work is equal to the product of that force you apply times the distance over which it's applied. So applying it to biology, and maybe most of you, I don't know, a lot of people have this somewhat misguided idea that biology is all about diffusion. Things just diffuse and find partners accidentally and things somehow work out. No. It's very deliberate. In fact, I would say one definition of life, there are many definitions, what is life, and we started with this, by the way, and you had one of the best answers to that, but another possible, so entropy reduction, right? Another definition of or distinction between living and non-living systems is that living systems have a deliberate directionality. If I want to move towards this gentleman, I can do that. A non-living system will not do it, will not decide. That's also the question, philosophical question of free will, which non-living systems don't seem to have any free will. We do. But behind this free will is also action. You have to exercise your free will to cause some kind of action. And maybe the most basic type of action you can think of is that of a motor protein. There's a lot of families of motor proteins. They're specialized to do different things. These are linear motors, so kinesin, dynein, myosin, actin-myosin complexes are what is used in our muscles. When we tense our muscles, these complexes strain up. There's also rotational proteins, FATPases, which rotate. So we have a lot of machinery of life that relies on this. That's why I'm bringing this up, because you need to fuel it. This is our biological gasoline, if you will, ATP or GTP. And so these motor proteins here, as you may know, probably all those people here who are from life sciences know this stuff inside out. but others maybe not. So, so that kinesin motor works by swinging. This is called a head, by the way. I don't know why biology is called feed head, but anyway, that's another. You know that, right? These are kinesin heads, motor heads. This, to me, is a head. It's a cargo carrying domain or something like that. Anyway, so these heads swing like that, find the binding site here. This one will find one here. And they take a step which is 16 nanometers in length. So this is delta L. This is the this is a microtubule on which kinesin walks. Dynein walks in the opposite direction. This one always walks from the minus end to the plus end. So it's a very directional motion. No mistaking. They unless you mutate them, then they will not make a mistake. And every step of that kinesin motor requires the usage of one ATP molecule. So it costs, let's say, 60 kilocalories per mole to make one step, which is quite amazing when you think about this. It's a lot of energy. So this is 1 ATP per step. It's really a quantum process this way, if you want to think in terms of quanta of energy being used up and transformed into movement. The force, by the way, which this molecule exerts is about 5 piconewtons. A piconewton is 10 to the minus 12 newtons. And as you may remember, 1 newton is 1 kilogram times meter divided by second squared. Okay. And you can start putting all of these numbers together. I told you about the power consumption of the human body. Last time it was 100 watts, right? 100 watts, which is 100 joules per second, and 1 joule is 1 newton times 1 meter. Anyway, these are just a lot of numbers. If you want to relate it to single cell and start counting how many such motors a cell can support at a given time, you can find these limits just by remembering that the human body has 3 times 10 to the 13th cells. There's a beautiful book I highly recommend for anybody who likes numbers and biology at the same time. It's called Biology by the Numbers, about 160 pages long, and it has all of these things. And you can spend a lot of time trying to understand biology this way. But this is not what I want to talk about. I'll just give you some examples. So these are biological energy quanta. And it's easy to remember, by the way. Somebody wrote a paper on this. And that converts into an easy-to-remember number, 10 to the minus 20th joules. That's the biological energy quantum, minus 20th. Okay. So we discussed in general terms why quantum mechanics would be nice to implement in biological systems. Some of these incredible things can be done in terms of quantum search algorithms that you can explore the entire space of states simultaneously, just like the wave function of an electron can go through all the openings and the diffraction grating at the same time, like a wave, rather than a single ping pong ball choosing and repeating and maybe this, maybe that. No, it knows right away. But there's a lot of objections to these ideas. I mentioned to you that most of physical systems which are operating at the quantum level are at very low temperatures compared to biological systems. Nonetheless, there are exceptions to it. There are, for example, nuclear spins of the phosphorus atom and lithium have been demonstrated to maintain quantum state for hours, which is what you'd like to see, because if we as human beings use quantum mechanics for some of our cognitive functions, maybe some of the and most brilliant minds use it more than others. You would like to have these quantum capabilities last more than a millisecond. But sometimes you have, I don't know, these are anecdotal things, people that have a so-called flash of genius, you know, a sudden insight, a bing, or, you know, it's a eureka moment, right? A eureka moment. Maybe this is an example where you really, that person's brain becomes quantum coherent and finds a solution which would be otherwise impossible to find, you know, over years and years. And maybe some of you also practice this or experience this, that you try to crack a problem for days on end and there's nothing happening, and suddenly you don't even think about it and you get the solution pop up in your head, right? Have you, anybody experienced this kind of thing? Yeah. Right. I did too, and I think it's great when it happens. Most of the time, unfortunately, it doesn't happen. So the first enemy of quantum mechanics is temperature, thermal excitations, because they destroy this, what we call coherence, or a well-defined state. You have too many superpositions and then the thing dissipates. And the second is, I mentioned already, wetness. So water, being water, dynamic, is not really good for maintaining quantum states. But nonetheless, and this is hopefully a growing list of physiological quantum effects which were either, well, they were demonstrated by physicists or biophysicists and published in high-profile journals. One of the earliest known to be examined this way is the power of the human eye. Our vision, sense of vision, is very acute. Experiments were done in total darkness and on subjects which were able to detect a single or 2 or 3 quanta of light. That's actually going back quite a number of years, maybe 50 years ago. That was already known. So the receptors of light in the eye, in the retina, rods and cones in the retina, are very intricately built, and they are able to receive a single quantum of light, electromagnetic radiation. And the question is, maybe to me, not so much that they are sensitive enough to register that and maybe undergo a conformational change. The question is actually of amplification. How this tiny signal gets amplified to the point that your brain and the entirety of your body understands or perceives light, which is amplified from single quanta, which are really tiny amounts of energy. So this amplification of quantum effects is, to me, equally mysterious as the effects themselves. This I mentioned also, olfaction. There's a number of papers on olfaction, sense of smell, where not the shape matching the geometrical fit of the molecules into the receptor site, but the vibrational dynamics are in resonance, leading to the recognition of a particular smell. Bird navigation is another well-known, if not well-known, but not really understood effect. You know, birds such as Canada geese every year make their migration in the fall to the southern United States and Mexico, and they come back in the spring. How do they know where to go? How do they navigate very precisely? This is really amazing. And there is evidence in the literature that they have tiny little magnets, spins, which respond to small deviations in the magnetic fields. So they can be fooled. If you change the magnetic field, they will. So they really respond to it. It's not some kind of collective memory or evolutionary imprinting. It is really a response to the magnetic fields. And it's very, very weak ones, yes.
Is that like a sixth sense that they have? Or?
It's not really even that. I think it is really a tiny little compass that they have. And the amazing, there's some literature I can point to that if you're interested on bird navigation using That causes the movement of the little compass causes electronic transitions, which cause the conformational changes, which then trigger the reaction of the brain of the bird to change the direction. So there is something really physical. It's a physical, amazingly quantum effect. And the amazing thing about it is that the tiny little amounts of energy, which would be actually quite a bit lower than the biological quantum, are sufficient to trigger response. There is a lot of sensitivity. And you have to really dig deep into quantum mechanics to explain how that is connected to these electronic transitions. Recent papers, well, last 7 years perhaps, in science, you can, if you Google quantum bird navigation, you'll find these papers. Anyway, that's interesting. Photosynthesis I mentioned a number of times, and I'll show you some pictures because that leads to some other consequences. And we talked about mitochondrial metabolism. I'll not come back to it. And finally, the most interesting and the most intriguing and the most controversial, consciousness, and that loops back into our brain, how we process. Well, first of all, the question is, where is memory in the brain? How do we maintain memories? How do we build new memories? How do we erase them? And you can do that. Of course, anybody who's been under general anesthesia understands the fact that we can controllably turn on and off consciousness without erasing memory, by the way, or to some degree. There is some deficit sometimes. Could this be an example of quantum biology? If you go to Wikipedia, quantum biology refers to applications of quantum mechanics to biological objects, and specifically they mention, I don't know if you can read it, some of these weird features of quantum mechanics, like superposition of states, that is being simultaneously in two or more different states, which classical physics doesn't allow, just like this electron going through both slits at the same time. Non-locality is this really weird aspect of quantum systems that Albert Einstein objected to. He called it a spooky action at a distance. That means that quantum objects, which are, let's say, prepared to be in a particular state, like spin up and two particles, one spin up, the other spin down, initially, they form a spin zero state because they cancel out. But you don't know which one is spin up, which one is spin down. And then you let them travel away from each other as if they were, let's say, ejected in opposite directions. And then, let's say, 1,000 miles away, you perform an experiment where you measure the state of one particle and you determine this to be spin up. Then the other particle will assume spin down because it mass cancel it without even doing anything. There is no action on the second particle, but it will be forced to be. In other words, it's almost like being entangled or shackled by the other particle. And that's called this spooky action at a distance. You act on one particle, the other one knows what happened to the first one, even though you didn't do anything to it. And the distance doesn't matter. It could be at the end of at the far reaches of the solar system, it'll still be that. And that has been demonstrated as well to be true. So it's really weird. There's no physical interaction, but there's a quantum entanglement. They are together forever. All right. Tunneling, yes.
This was demonstrated through an experiment? Or
yes. There was. Well, you can do experiments, maybe not over millions of kilometers, but we have a few, let's say a few thousand kilometers. You can set up an experiment where you prepare two particles. Okay. The limitation is you have to shield these particles from external influences. So, in other words, we couldn't do this in an environment like here or anywhere where they can be colliding with other particles of ultra high vacuum and shielding from electric and magnetic fields. So I don't remember what was the distance, but physicists create such conditions in accelerators in like Geneva. I don't remember where it was, maybe in Geneva and CERN. And then you can demonstrate the validity of the collapse. It's called the collapse of the wave function of the system.
But like that level of control is not there in biological systems.
Yes. And this is obviously a valid point, that biological systems are never isolated like this, or almost never shielded, because by definition they cannot be. If you were to isolate a biological system in a vacuum, that system would die very quickly. You have to be pumping it. However, there may be some, and this is actually a very, very long-lasting and robust discussion along these lines. Some degrees of freedom may be shielded to some degree, and that's people are searching for such things. For example, the nuclear spin is largely nuclear spin of particles such as phosphorus is largely shielded. It doesn't interact with much, if anything, and these you can create such large quantum states that exist, being surrounded by other particles, but they still do not interact, and that's very specific cases. It's called an S state. It's net spin zero. So very exotic, nonetheless not completely impossible. And the question is, if it's not completely impossible, is it manifested at any point, at any in any case? All right. So this is as opposed to these trivial applications of quantum mechanics to quantum chemistry. As I mentioned to you, chemistry underlies all of biology. So by definition, you could say that all biology is quantum. But yeah, this is in a trivial sense, that yes, we make these bonds, but that's it. All right. Let's just maybe just another historical remark before we go into back into the neuroscience aspect of quantum biology. I just want to mention that that most of these early developers of quantum mechanics thought about this, and they thought about biology. And I mentioned to you Schrödinger, who wrote this book, What is Life?, that I highly recommend you pick up and read. It's 1943, I think, so it's 75 years old, still very current. But people like Bohr, Heisenberg, John von Neumann, the creator of modern computing, they thought about this. John von Neumann actually provided an estimate of the size of the human memory, how much memory can be stored, how many bits of information. Hopefully we'll talk about this in a few minutes, in the human brain. Carl von Weizsäcker, I mentioned Elsasser, Weishaupt, Eugene Wigner, Freeman Dyson, who's actually a very interesting character, a very brilliant guy. You can find some YouTube videos with his opinions on many topics, and others. So this is not a topic restricted to people sort of from the last few years, but also talked about for decades, going back to the beginning of the previous century. Okay, and much of it has to do with also with photons. In a quanta of electromagnetic radiation, can photons or biophotons be instrumental in it? We know that they exist, biophotons. This has been demonstrated almost a century ago by Alexander Gurevich in the then Soviet Union, in Russia. And you may have heard about images of human bodies emitting photons, or aura, giving an aura. This is related to our metabolism, our being alive. And it's beyond the thermal radiation that we talked about. Black body radiation is something else. Everybody at a finite temperature emits infrared radiation, and a spectrum which is well characterized and explained by Planck. But biophotons is something else. It's photons which do not conform to that spectrum. They are so-called non-thermal. They are emitted as a result of metabolism, our biological activity. And I think I have the next slide, which actually segues to it directly. And if you have, again, a little bit of time on the weekend, you can Google this. The website is called Cell Intelligence. Cell Intelligence. And this website illustrates the body of work due to Günther Albrecht Bühler from Northwestern University in Chicago, who spent about, I don't know, 25, 30 years researching this topic of cell intelligence. What a single cell exhibiting features of intelligent behavior. And he specifically, so this is a cartoon kind of illustrating schematically how he sees the cell. Of course, the membrane, which is the outer limit of the cell. You have the cytoplasm with the cytoskeleton, the machinery that propels the cell, actin filaments, microtubules, motor proteins, all of this allowing the cell to move, which is important in this connection. And then he pointed to the structure which is sometimes called the greatest enigma of cell biology, namely the centriole. I don't know if you ever thought about this. The centriole is a tiny organelle, about 500 nanometers in size, comparable to mitochondria in size, but it's composed of 9 triplets of microtubules and a central axoneme, a doublet, surrounded by some proteins of various kinds. And this is the first structure which undergoes cell division, duplication, centriole duplication. Actually, two centrioles form a centrosome. Right? They're oriented perpendicular to each other, two centrioles. And when the cell divides, these float away from each other, basically. And each grows another partner. So then it's duplicated, and then they move to the opposite ends of the cell. And from that, microtubules spring, form mitotic spindles, and the spindles reach out to chromosome, and that's a cascade of events that leads to cell division. But the very first thing that happens is duplication of the centrosome, or floating away of the centriole. So that's an enigma in its own right. But this is not what Günther Albrecht Bühler is talking about. He's talking about something else, namely, implies the centriole is the eye and the brain of the cell, because it's the structure that is sensitive to signals from the outside. He did a lot of experiments, and you see some kind of short video. He tried to demonstrate that that cells perceive other cells in the neighborhood, and if these are cells of the same type, they will move towards each other. And he tried to figure out if this signaling, because they have to perceive somehow, that there's a cell, let's say, 20 microns away, how do they know it? And it could be, of course, by mechanical means, maybe if they were touching, but he made sure that they were not touching by any structure. Secondly, by chemical signaling. So he eliminated this by putting some barriers between them, quartz. I think they were in 2 different quartz dishes, containers. And they were still perceiving each other. So then he figured out if they are not touching or sending chemical signals, any material signals, there must be electromagnetic means of interaction. And what he did was he then proceeded to find out at what frequency the cells would be sensitive to it. In other words, a source of electromagnetic radiation, a particular frequency, would mimic the behavior of another cell. And this is a reflecting bead, and he was using tunable laser light, and he found these peaks around 900 nanometers to 1,000. So that was basically the range over which there was a sensitivity. The cells would respond to the incoming light and move toward that. Actually, it's somewhat visible. Some of it is visible up to 750, but some of it is near infrared. It's not visible light, but in the near infrared region. this is the structure of the centriole, and he concluded by observing the movement of the centriole that centriole is the organelle, the structure within the cell that is sensitive to light and transduces that electromagnetic signal into a cascade of electronic and mechanical transformations, which eventually result in the cell moving toward the source of light. So that would be the most basic aspect of intelligence, being able to observe and react to a signal. Okay, and the way, it's not really quite clear how it is mechanically achieved, but he speculates on this website, and you can find a lot of material there, that these triplets of microtubules actually move or rotate toward the source of light and can detect the direction in both in plane and out of plane to within 3 degrees. So very precisely the origin of that light. So that's kind of interesting. All right, so there is something there. And then very briefly, let me just go through this evidence for quantum coherence in photosynthesis. So everybody knows the biological meaning of photosynthesis. Basically, the most fundamental source of energy on this planet. Therefore, it's quite important, but it's involved in the leaves, not only in plants, but also there are some bacteria which are photosensitive and algae that are able to convert light energy into electronic transitions. Chromophores are the structures where this takes place, and importantly, these are molecular structures with separations between units on the order of 15 angstroms. That's important because 15 angstrom is more or less the limit for quantum tunneling. If it's more than 15, it's called the first order distance, 15 to 20 angstroms. Beyond that, you cannot do this quantum tunneling. And so you have light energy being absorbed by light harvesting complexes, and light harvesting complexes transfer this energy to photosynthetic reaction centers, and then they can store this energy, and some of it can be converted into ATP, once again biological energy quantum, and the remaining can be used to remove electrons from water molecules, and so on. That's a cartoon of that called FMO complex, where things happen in terms of photosynthesis. And if you look more closely, the chemical structure of these complexes, it's called the FMO complex, as I mentioned, consists of 7 rings, heterocycles actually, arranged like that. And as I said, the distance between these rings are within 15 angstroms. And that's sufficient to create a coherent quantum state for the entire complex of 7 multi-ring molecules. And these were probed by laser experiments, and it was demonstrated in the last decade that these complexes create a quantum entangled state. They have a quantum wave function that extends over this entire grouping of 7 multi-ring complexes. This is the work of Engel et al. From 2007. Used electronic spectroscopy to study these electronic excited states of electrons in the complexes, and concluded that this is a schematic actually of this whole experiment. They use 2 lasers and observe something called quantum beating. Quantum beating is analogous to classical beating in acoustics. You may have heard about beats. When the radio is a little bit out of tune, you can see kind of the amplitude going up and down. That's basically 2 frequencies close to each other superimposed. And then you have the in and out of phase oscillations. So the same thing was observed in these complexes for electronic states, and described by physicists who created this quantum Hamiltonian for the 7, and these are the distances and these are the energy parameters for the Hamiltonian, and the theory was created, the model solved, and frequencies matched to those observed. Anyway, it works. So great. I mean, it's now pretty much accepted that photosynthesis, chromophores in plants and FMO complexes in algae and some light-sensitive bacteria use it. Is that the end of the story? When you look at this complex, and I'll show you another one, you can see some similarity. Not only similarity, but the parameters, these numbers are the parameters of the Hamiltonian, are very similar. This, by the way, is a completely different structure. This is tubulin. Tubulin, and these complexes here are tryptophan molecules, amino acids of tryptophan. Tubulin dimer is a building block of microtubules, and each monomer, alpha tubulin, beta tubulin, has 4 tryptophans. And they're arranged in such a way as here that the distances, again, between the neighboring tryptophans are within 15 angstroms or so, which means that they also can create a quantum complex, and the electronic degrees of freedom for these tryptophans can be shared, or they can create a coherent superposition state. So back to your question, that kind of answers. It has not been directly demonstrated that microtubules can be excited quantum mechanically and a collective state, quantum state, can be created. But hopefully it'll be done soon. But there are some indirect consequences of it. I'll tell you about in a few minutes before the end of this class, which indicate that this is happening. So yes, this is not done in vacuum. They are not isolated, but they may be sufficiently strongly coupled that external influences do not matter that much. And in fact, sometimes if you talk about tunneling or transitions of some quantum particles, a little bit of thermal oscillation helps. If you have to overcome a barrier, jiggling it a bit helps to do that. So this may be the case. In other words, there may be a sweet spot. Sometimes, you cannot simply expect total shielding and vacuum. Yes, this would be ideal, but sometimes actually a little bit of thermal excitation helps. There's thermally excited transitions, for example, in some catalytic reactions, which help. So again, I'm cautiously optimistic that this is one of the systems which will demonstrate the existence of quantum signaling transitions in living systems. Why would this be important? Okay, and so I don't want to blow my own horn, but with my student we did the calculation and we demonstrated that this can be quantum sufficiently long-lived state, not hours, but nanoseconds. So it's a good step, first step. And this is the interaction Hamiltonian between the dipoles, transition dipoles of the tryptophans and tubulin. And these are the simulations showing you have this quantum state decays or becomes thermalized, dissipates over a nanosecond. We would like to have it, longer lived, not maybe a second or tens of seconds. You don't have to have it stable forever, because these states can be repumped by energy. As we talked about today, biological energy is in great supply, so you can make transitions to excited states generated by the supply of energy. So Travis Craddock is the student who did all the calculations. He's now a professor in Florida. And this is how you extend it. I showed you a complex of 8 molecules, 8 tryptophans, but now this is one tubulin dimer. Microtubules are formed of 13 protofilaments of tubulin. I don't know if you remember from biology, but that's a microtubule, and this is a tubulin dimer, alpha, beta, and there's 13 of these protofilaments making one microtubule, and then you may have up to, let's say, 1,000 or 2,000 rows, which would give you a microtubule that is 10 to 20 micrometers long, which is typical in cells, including neurons. And neurons you have, as you will see in a second, neurons are packed with microtubules. So what I'm trying to say is that I showed you in the previous slide only one of these units, and you have 8 molecules, tryptophans, that can be isolated for this quantum state. But if you build a structure like this, you'll have tens of thousands of these forming an array, almost like a quantum antenna that can be vibrating. And this is kind of a representation of these. It's one patch. So let's say I'm showing you here something like this. But you can extend mentally, at least, this almost to infinity, because you'll have thousands and thousands and wrapping around. And these will be the pathways through which you could send quantum signals. If you want to use it for quantum signaling, quantum information processing, if you will, if this is your quantum computer in the brain, yeah, you can think about it like that as well. All right, and where is the energy coming from? Well, as I mentioned, GTP, but it's delivered slowly. There is actually a much greater source of energy in the ultraviolet range from recombination of reactive oxygen species, which is happening all the time as byproducts of mitochondrial function. So energy can be delivered and used to excite these transition dipole states. So now we can start fantasizing. This is the future of medicine, right? So we can now look into the future. So far I've talked all about the past and the present, now a little bit about the future. There can be almost an unimaginable number of things happening that can involve these structures in some kind of computing, signaling, memory storage. Because of the dynamics of microtubules in all cells and specifically in nerve cells. And the complexity of this is cosmic. It really is astronomically complex. Number one, there's alpha and beta tubulin, each of which in the human body exists in about 8 isotypes. Every cell of the body makes it differently. There's 20 genes coding for tubulin, and every species has a different sequence. And in every cell, there are also post-translational modifications which change the surface of the tubulin by glutamination. Some other additional groups can be attached to, especially C-termini of tubulin. And, of course, you can also have astronomical number of mutations. So if you start thinking about this as potential manipulation of biological manipulation of these systems for specific purposes, there is no limit to your possibilities. Okay, and then microtubules, maybe I should show you some, because I have some nice slides. So of course, we talked about this, cell division. This is a neuronal cell, and the solid bars are microtubules. They're interconnected by microtubule-associated proteins, MAPs, especially MAP tau, MAP2 are the main ones. And almost everything to do with our cognition, an impairment of cognitive function has to do with microtubules. There's almost no exception. Every neurodegenerative disease is rooted in problems with the cytoskeleton. Microtubules are motor proteins, most importantly microtubules. For example, Alzheimer's disease is at least half of the community in Alzheimer's disease research believes in tauopathy, which means hyperphosphorylation of tau, MAP tau, which disconnects microtubules and causes the degradation of this network and eventual death of neurons. So loss of memory is happening on the way, and therefore that may give you some clues where memory can be stored. Traumatic brain injury. Hockey players, football players, they suffer from this a lot, concussions. And post-mortem of many of these athletes demonstrated complete correlation between the pathology and the breakage of microtubules in the brains of these players. So that network is a very good candidate, to say the least, for memory storage, information processing, signaling, and some kind of integration of cognitive functions. Examples, actually I co-wrote a book on this, so if you Google just on this topic alone, it's called nanoneuroscience. There's a lot of information about these various connections. These are correlations, not causations necessarily, but you have to start somewhere. You may have heard, just in passing, the work of Roger Penrose, one of the greatest physicists alive from Oxford, and Stuart Hameroff, anesthesiologist at the University of Arizona. Over the past 20-some years, they developed this model that consciousness actually occurs at the level of microtubules in the brain. That's the substrate for consciousness. It's actually a bit deeper than just cognition. There's the so-called qualia, concepts which are not just like reaction to a stimulus, because we can think about receptors and machines that can react to stimuli. It's it's controls. If it's too hot, you turn down the heat. If it's too cold, you turn it up. It's not that. It's deeper. Qualia is concepts that we have in our brain, such as redness. What is redness? The concept of the perception of color, red or black or yellow or things like this. Deeper aspect of our cognition, abstract concepts. So anyway, that's the most profound, if you will, understanding of the brain's utility to us, not just as an information processor and a control center, but also an amazing, powerful organ that allows you to develop abstract thinking and be creative. To the best of my knowledge, artificial intelligence has not been creative yet. It's whatever you program, you train an artificial intelligence algorithm, it'll try to find patterns, but it's not going to create anything new. Our brain is capable of doing this, and how does this happen? So anyway, this is just to give you a taste of this connection. And very briefly and quickly, so microtubules are not only the most prominent parts of neurons, but they also interact with ion channels, so they allow communication between cells, affect or regulate, if you will, ion channels. And through actin filaments, they participate in axoplasmic transport, and that includes neurotransmitter activity and transport of RNA to the synaptic receptors. So the challenge is how to create a logical hierarchy, starting from this humble protein tubulin to a microtubule, to a network of microtubules integrated by MAPs, to a dendrite. So most of neuroscientists are telling us that most of our cognition takes place in dendritic areas of neurons, not so much in the axon, but in the dendrites. And dendrites have a very interesting property, namely they have microtubules which are bidirectional. That's the only part, in fact, the only place, to my knowledge, in any cell where microtubules are oriented antiparallel, which means one microtubule will be going from minus to plus this way, and the other will be going next to it in the opposite direction. Why would that be? And probably, if there is any functional significance to it, it is because of the feed forward and feedback process. So maybe you can send back a message to the soma in the neuron that, yeah, we got it, don't send us any more RNA, we have enough, or something like this. Back propagation. But looking at, so I talked about tryptophan excitations, I talked about electron hopping, so electronic transitions. I didn't talk about C-termini. Tubulin has also C-termini, which are really antenna-like objects, so peptide sequences, right? So a microtubule is not really a cylinder. It looks like a toilet brush because it has these tails, C-terminal tails, which are, by the way, very negatively charged electrostatically. So they are sensitive to electric fields, to magnetic fields, etc., electromagnetic fields, and maybe for a reason. Conformational changes, so structural, much slower. But if you were to look at all of these things separately as channels of communication, just like your cell phone may have one channel for email transmission, another for web browsing, another for calling, another for, I don't know, something else, beeping, acoustic signal. You have 4 channels of communication in a microtubule, and therefore you could have a lot of possibilities. I did a calculation of how many bits of information this could store, and you'd be surprised. If you count the number of dimers and the number of microtubules in a single neuron and the number of neurons in the brain. By the way, what is the number of neurons in the brain? Does anybody know? How many neurons do we have? 100 billion on average, yes. 10 to the 11th neurons in the brain. And there is, each neuron has about 100,000 tubulin dimers. So if you do the math, if you make a product of these two, it's 10 to the 16th units, processing units. Each unit may have 4, at least, maybe more channels of communication. Each of these channels may have several bits. So this could be maybe 10 to the 18th bits or more. Is this a lot? How much? Okay, we are happy with gigabytes, right? Or terabytes? Terabytes. So your hard drive may have it. Maybe not your hard drive, but my hard drive. So 1 terabyte is 10 to the 12th. So we can have a million terabytes in the brain if we use all our potential, which is a lot. It's like maybe not all the computers in the world, but pretty much close to it.
So you talked about the tryptophan excitation and like how tubulins could have this quantum
Yes.
going on because of tryptophan.
Yes.
A lot of all the catecholamines that are the neurotransmitters that are there for like higher brain function, reward stuff, and like avoiding punishment, they're tryptophan based. Could they be like
Absolutely.
interacting?
Yes. Yes, I think so. So this is a field which is very much dynamic and evolving, and we're just looking at the foundation. Interactions with other structures in the brain, very few people understand. I'll give you one anecdote in the interest of time. The European Union invested about €1 billion, which is $1.5 billion, in a mega project, a so-called flagship project called the Human Brain Project, HBP, in Lausanne, Switzerland. And so with 1 billion euros, their ambition was to link together 1 million processors to mimic or to create something like an artificial brain. So that the scale, and that's more or less the most ambitious thing that Europe has ever done in recent decades. So that means that their objective was to represent 100,000 neurons by one processor. I met with a few times actually with the principal investigator Henry Markram who did this. I said, but the neuron itself is already a very intelligent device. You represent 100,000 neurons by 1 bit. Are you crazy? No. No, they got the money. I didn't. But what I'm saying is that maybe it would be better to spend €1 billion understanding one neuron and what's happening inside a single neuron. And then you can scale it up and see, okay, what happens when you have two? Maybe these other molecules, neurotransmitters, send signals between neurons and something important is happening inside. Anyway, you can see the scale, right? So when you, from this point of view, one neuron, based on our calculation, has about 1 gigabyte of memory available to it. So it's like an old-fashioned memory stick. Now, probably memory sticks these days have more than a gigabyte. But a neuron is micron, micron dimensions. Okay. So these are the big, deep questions that a lot of people are trying to answer. We don't have really any good answers to those. What makes the brain special? I told you, I mean, the incredible potential to store memory and to manipulate information. So now the question is how fast and how much can you process? If you have, let's say, 10 to the 18th bits, and you can switch them. Now the processing speed. What's the processing speed? Some of these degrees of freedom that I mentioned can be processed very fast. They can be processed at a nanosecond. Typical changes at the protein level. But this is too fast because that would be 10 to the 27th flops or something like this. So operations per second. I mean, that's probably more than the entire universe can do at present. So anyway, we have some oversupply issues. There's just too much that this can do. Where is memory stored? I have a few minutes. I'll show you where we think it is. What is the computational power of the brain? Well, it's quite amazing. This may be overly optimistic. Of course, using all the degrees of freedom all the time at these super speeds, maybe it's not feasible for various reasons. But it's still quite impressive, as you'll see. And is information processing in the brain most important, I think, because I've been still talking to you as if it was kind of like a linear object. No, it's actually hierarchical. There's a structure, very interesting structure, and maybe it's something like a fractal or hierarchical, a structure within a structure within a structure. Maybe fast degrees of freedom feed into slower at the larger scale and even slower at a bigger scale. And eventually as you may know, but our so-called pre-processing time, it is about 500 milliseconds, half a second to react to anything. That's the fastest we can react, at least to on average. It's hard to explain how fast, let's say, athletes react, like hockey players, McDavid and so on. It's probably much faster than even a millisecond because the speed at which these things happen and how you have to react. It's almost beyond, I think not almost, I would say beyond explanation using sort of classical physics. And the time it takes for the signals to travel down the neuron and back or from the optic nerve to the action of the hand or some other part of the body. I don't know if anybody has actually done these calculations, but I'm quite sure that things like tennis players, when they serve at 160 miles an hour and the opponent gives a perfect return, can this be explained by mechanistic calculations? Because these are split-second things. The same with hockey players. The same with baseball, right? The speeds are incredible. Okay, and then I kind of mentioned to you that all of this, even at the most conservative estimates, uses incredibly low energy because we use something on the order of 1 to 10 watts for the processing of the information in the brain. So cognition costs us almost nothing. Yet, the power that the human, okay, this is actually given here. The power, so I said 25 watts for the entire cost of running the brain. Most of it is actually for trivial things, maintenance. But the processing is maybe 10% of that, about 3 watts. And the human brain can compete. Maybe most of the time we'll lose, but can compete with the blue gene, one of the most powerful supercomputers on the planet. The blue gene consumes 1.5 megawatts of energy. Our brain consumes about 3 watts, so about 1,000,000 times less. And we can play chess with the blue gene, and I think Kasparov, I think, kind of tied. I don't remember exactly, lost in the end, but anyway, it's a fair competition, or Jeopardy or things like that. All right, so one more thing that I forgot to mention in this connection is that each neuron interacts with, on average, 10,000 other neurons. So that's the interconnectedness, and some actually interact with 100,000, which is quite amazing how all of this is integrated and organized. Okay, so these are back to this slide. Estimate how 1 gigabyte per neuron, 100 kilobytes per microtubule. With 100 billion neurons, you have 10 to the 20th bits per brain. Now, at microsecond transitions, you end up with 10 to the 26 operations per second, which is way more than the blue gene supercomputer. The best supercomputer in the world doesn't have this kind of operating speed. 10 to the 15th, actually. So that may be an overly optimistic assessment, but even if you just say that we operate at a microsecond transitions or millisecond, you're still better. The only thing that, of course, throws this calculation out of whack is that we say we don't use more than a tiny fraction of our brain, and probably that's true. So we kind of restrict ourselves most of the time to and limit our capabilities. What happens if we don't? I think then there's some incredible things happening when people utilize their brain to a greater degree. This is the okay. I think we are running out of time. I just want to show you one more thing in terms of what is memory and where can it be. So a lot of ideas about where memory is stored. Some people believe it's holographic, so our brain creates a hologram of the universe. This is the work of Carl Pribram. Some people believe in this fractal resonance structure. There's even more exotic ideas by Rupert Sheldrake. He wrote some very interesting books, a biologist from Cambridge. He says that memories are not stored in the brain at all. Actually, that there is something like memories are not in the brain, but in the universe, and we only access it, almost like downloading from the cloud. You know what I mean? I think now we can actually explain it better with this computer lingo, that there's a cloud for the universe with all the memories, and you have access to your memories by downloading or accessing it. But that's some crazy experiments showing that there may be something to it, that the memories are not only in the brain. Of course, yes, physiological experiments demonstrate that we do have memories in the brain. Anesthetics turn off the consciousness. You can lose memories by accidental damage or molecular damage, Alzheimer's. I just want to mention to you, to throw some kind of curiosity into it. There's a scientist called Mike Levin at Tufts University in Boston. He did experiments with worms that have brains, and he decapitated these worms. They regrow their brains, and taught the worms to solve some puzzles and mazes. And after decapitation, they remembered how to solve them. So just to tell you that maybe at the level of the human being, all we know is our brain for memories, but maybe at the lower level, other organisms use other parts of the body to record information. So this has been discussed for a long time, and people diverge in their opinions how much memory we have. I'll just show you one thing. There have been experiments with mice that allowed to erase temporarily memory in mice by using inhibitors of calmodulin kinase. It's an enzyme that is activated by calcium in neurons. So calcium influx causes the assembly of calmodulin into a hexagonal hollow enzyme, and it phosphorylates microtubules. And so it means that this enzyme, it's a motor actually also, but very complicated. I'll show you the picture of this. Here. This is that enzyme. It looks like a nano robot or what do you call it, transformer. Walks on microtubules and phosphorylates these tubulin dimers as it walks. So it turns out that if you inhibit this enzyme, mice at least lost memories. Therefore, the idea is that memory in this particular case is just encoding on the surface of neuronal microtubules a pattern. This pattern is pattern of phosphorylation sites. This is a picture of the calmodulin kinase 2 walking on a microtubule and leaving a trace. This is reversible. You can dephosphorylate and erase the memory as well. Anyway, so that's an idea, and if that's the case, you have another huge potential to add another channel, maybe on communication, but special channel for information encoding in the brain by using enzymatic reactions. And this can lead to the creation of logical gates and information processing also by rearrangement of maps that interconnect microtubules. So I will stop at this point because the time is up, but I'll tell you something about the midterm exam that you probably want to know. There will be some questions, right? Yes? Okay, so there will be 7 questions, but you choose 4. Choose only 4 questions out of 7. Right? You got it? And there will be descriptive questions. Something like, what is the main difference between quantum mechanics or quantum physics and classical physics? Or why would quantum physics offer something useful to biology? Or what are the possible objections to quantum physics playing a role in biology? This kind of thing. I can't give you more, but you get the picture, right? So hopefully, if you didn't take notes, you can watch the video again, which will be posted. But this will be kind of descriptive. So list a few examples. It's just this kind of question. And you'll have 4 to choose out of 7. So there'll be, if you're not comfortable with something, you can always find something that you like. All right, any questions to wrap it up? No? I'm sure it was a lot. As Dr. Solis said, we add lectures to it, and now it's from 1 to 2, now it's 3. Next semester I won't be giving it because I'm on sabbatical, so I will not be in Edmonton in the fall, but maybe a year from now. If you have any suggestions how to change it, or add to it, or remove from it, let me know. Thanks for your time.